So good day, everyone, and good morning or good afternoon, depending on where you are in this world. Before I start, I just want to give a few housekeeping details. First, that this meeting will be recorded, so keep that in mind. And also, I'd like just to say that the content of this seminar is entirely the responsibility of the presenters and does not necessarily, is not necessarily consistent with the views of FAO or its members. I don't want to take a lot of time, but just want to give a brief introduction to give some context to today's presentation. Since 2009, the Iowa State University and FAO have been cooperating on the university's Dean's Global Food and Agriculture Leadership Program, which is part of their study abroad curriculum. In this program, a group of students and professors agree with staff members of FAO on topics and a project to undertake. Then, in the spring semester of each year, starting, say, January, February, the students start on the project, and through this class also are informed on other aspects of food production on a global level, and then they work on the project up until the end of the spring session. In May, the course culminates with a trip to Rome for about 4 weeks, where they can work directly with FAO to finalize the project. So I've been fortunate to work in this, collaborate with this program for several times over the years. So I was very happy when I was contacted by Stephen and Elizabeth Lonergan, the supervisors of this year's group, who asked me to participate again this year. Of course, I just needed a topic which was of interest both to us and to Iowa State and the students. Therefore, I discussed with my colleagues, Gregoire and Rosvita Baumum, who are here today, as well as with Christian Luft, who is an ex-colleague, was a FAO fellow who was based, and I guess was based before, at the University for Applied Sciences in Neubrandenburg, Germany. At the same time, we started preparation for the next report on the state of the world's animal genetic resources for food and agriculture, which is a major publication which we do every 10 years. This publication includes some in-depth analyses or studies on topics that are of present and future interest in the management of animal genetic diversity. We got feedback from countries that both management of animal genetic resources in the context of climate change and the use of new technologies, including genome editing, would be of interest for the countries and then for inclusion in the state of the world. Therefore, this gave us the idea to take advantage of this opportunity to propose these ideas as a topic for the work. Luckily, the students and the advisors agree, and the project, therefore, is a literature review on these two topics that will serve as the basis for subchapters within the eventual State of the World document, which would be scheduled to come out in 2027. So without further ado, you have the background now. I would like to introduce Dr. Carmen Bain, who is the Dean of Academic Innovations at Iowa State University, who will introduce the student group.
Well, good morning, everyone. And on behalf of the College of Agriculture and Life Sciences at Iowa State University and the faculty leaders of this program, both online and in person, who will introduce themselves shortly, I am delighted to see so many people here, both virtually and in person, to hear the results of our students' hard work all semester long. So thank you all for being here. I would like to elaborate a little bit on Paul's really nice overview of this program. The Dean's Global Agriculture and Food Leadership Program is a really special program at Iowa State that's organized out of the College of Agriculture and Life Sciences. Undergraduate students from any major can apply and are then selected to participate in this program. And what's unique about it is that students get to work on a real-world project with experts at FAO. That's a pretty unique opportunity. And in addition to working with our colleagues and experts at FAO, they do this work under the guidance of faculty experts at Iowa State. And as Paul said, they began working on this project in January. They meet twice a week for 4 hours all semester long. And in addition to thinking about, what are the big global issues in relation to food and agriculture? What are the big problems we confront? They also learn about the UN system. They learn about the work of FAO and the SDGs and how FAO as an organization engage in these issues. So they learn about the content, but what we want is our students to develop as young leaders and problem solvers. So the content is important, but we also train them to think about how to become leaders, how to work in high functioning teams, how to develop their communication and research skills so that they go out and they help solve these issues. Midway through the semester, they start collaborating with Paul and his team, as Paul mentioned. When they come to Rome, they meet with FAO, they come here, they collaborate more deeply, but we want them to be in Rome too. We want this to be an in-person experience because we want them here to learn about the history and the culture, the architecture, food systems of Rome and Italy. So that's also an important part of the experience. So before I go on, I would like to have each of the students give their name, their major, and the year that they're in at school. Amy.
Hello, my name is Amy Taylor. I am a junior at Iowa State, so I'm in my 3rd year. My majors are in animal science and global resource systems. Hello, my name is Jillian Anderson. I am a 3rd year student in agriculture and rural policy studies. Good morning, everybody. My name is Jennifer Holliday. I just completed my 2nd year at Iowa State, so I'll be going into my 3rd. And I am majoring in agriculture studies, journalism and mass communications, and international agriculture. Good morning, everybody. My name is Abby Sesker. I'm going into my 4th year at Iowa State University, and I'm studying animal science with a minor in meat science.
Hello, good morning. My name is Rebecca Larson. I am going to be a 4th year studying ag policy, international agriculture, and political science.
Good morning, everyone. My name is Shraddha Siluri, and I'm going to be a 3rd year student majoring in global resource systems and environmental studies.
Hi, everyone. My name is Molly Simmons. I'm going to be a 4th year undergraduate student in environmental science, global resource systems, and soil science.
And could we have the faculty introduce themselves? So we'll begin in person, and then we have Joe Coletti and David Acker online, and they can introduce themselves afterwards. Elizabeth, would you like to start?
I am Elizabeth Lonergan. I'm Elizabeth Lonergan, professor in animal science at Iowa State University.
Hello, I'm Shelley Taylor. I'm the Director of Global Programs for the College of Agriculture and Life Sciences at Iowa State.
Good morning, my name is Stephen Lonergan. I'm a professor in the Department of Animal Science at Iowa State.
Joe, can you unmute and introduce yourself?
Yes, good morning, everybody. My name is Joe Coletti. I was the Senior Associate Dean at the College of Ag and Life Sciences. Have been engaged with this fine program since the beginning, and really enjoyed all of the interactions with FAO and the students and faculty over the years. Thank you.
David?
Hello, everyone. David Acker, Associate Dean for Global Engagement. Looking forward to the presentation.
I think it's really important that the faculty and students introduce their majors, because it's really intentional that we want students who are coming from different disciplinary backgrounds, and that's one of the things we really appreciate with FAO, and one of the big takeaways that students gain from working with you is that to solve complex problems, we need to be bringing together teams of people from different perspectives and different disciplines. So I think that's really valuable. So on behalf of the faculty and the students, I want to extend our gratitude to Paul and his team. I've heard wonderful things throughout the semester about just the amazing mentorship that the students have received from each of you. You've given up your time. This is folks from FAO going above and beyond. This is not your day job. So you spend time with the students, you help them think about the problems, you guide them in asking the right questions, how to conduct the research, you help them by posing critical questions and pushing back and helping them think really deeply. Yes, Gregoire. Engaging really deeply about this. So, and I especially want to thank Paul Betcher, who, as he said, has been with this program since the beginning, and we couldn't have a bigger advocate, friend or colleague than Paul, and really there are no words to express our gratitude for Paul. And finally, the last thing I'd like to say, as you think about what the students present, we're incredibly proud of what they have accomplished. They're undergraduates, and as you heard, some of them are second year, what they've accomplished, but I also want us to walk away and think about the bigger impact this collaboration is having on these students. Yes, the students have accomplished a lot through working on this project and what they've learned about genome editing and phenotypes and livestock, but the bigger thing is they've learned about how do we think about these problems? How do we collaborate? How do we think about these problems not just as scientific and technological problems, but these are problems that are deeply social, political, economic, and we have to be bringing together these different perspectives and voices. And this is what we learn from our collaboration with FAO. So thank you, and thank the students. So now I would like to invite the first group to begin.
Good morning, everybody. My name is Jennifer Holliday. The team I am a member of and myself will be presenting to you all on genome editing in livestock. Alongside myself on the team, there is also Amy Taylor, Jillian Anderson, and Abby Sesker. As you just heard Dean Bain mention, we all come to you from Iowa State University and have been partnered with FAO throughout this past semester. About 3 months ago, we were presented with this project and have been doing intense research ever since. Today, throughout the rest of our presentation, we will be discussing objectives and methods that we use to gather all of this information, climate change impacts around the world, the history of genetic selection, transgenic and genome editing and the differences between the two, genome editing and examples, as well as potential challenges that this may present to the world, producer and consumer willingness to adopt. So how are producers and consumers going to move forward with the idea of genome editing? Policy and perceptions, as well as the future and potential outlook of genome editing. The objectives that we took into consideration when we were presented with this challenge are, as you can see on the screen. We really thought about the sustainability development goals around the world, especially looking at human livelihoods and welfare, animal health and welfare, environment, and food security. We also took into consideration the opportunities and challenges that genome editing can present with us, and finally, especially, how genome editing can impact low and middle income countries worldwide. When it comes to our methods, we did an intense literature review throughout the past month while we were in Rome, and even began when we were back in Iowa. We also had many guest lectures while we were still in class, including Dr. Jack Decker's PhD candidate Blythe Schultz, as well as Dr. Juan Steibel. We also continued to do an analysis of the SDGs and did interviews once we came to Rome with field experts such as Mike McGrew and Katrine DeVolder. We also have done weekly meetings with our FAO colleagues, including Dr. Paul Betcher, Dr. Gregoire Leroy, Dr. Roswitha Baumung, Dr. Christian Luft, and Rama Jalab. Moving into climate change, as we are all aware, our world is in severe impact of climate change that we are causing. This includes rising temperatures, increasing CO2 levels, disease prevalence becoming higher and higher, a decreasing availability of water resources, and even losing prominent animal and plant organisms. This all comes down to SDGs, as you can see on the screen, and can negatively impact our food and water resources around the world, which is what we will continue today as I invite Amy Taylor to come and share an introduction to genome editing.
Thank you, Jennifer. Genome editing is the process of making subtle changes to the DNA using biotechnologies. As you can see from this diagram, once scientists discover a gene of interest, revolutionary technologies such as CRISPR-Cas9, zinc finger nucleases, or TALENs can create a double-stranded break in the genome and allow editing in that specific spot. So our team wants to make it very clear that today we are not talking about transgenics, we are talking about genome editing. Transgenics is the introduction of foreign DNA. That foreign DNA could be in the form of the same species, different species, or synthesized DNA created in a lab. What we are talking about is genome editing, which is the edit of the original DNA itself. Because it's the original DNA, it has the possibility that it could potentially occur naturally. Now that those terms have been defined, I would like to talk briefly about selective breeding. Selective breeding is a practice that has been going on for a very long time, even before the term was created. It involves breeding of plants or animals that have selective traits that will produce offspring with favorable traits. So what I mean by that is if you were to breed a Holstein cow with high milk yield with an Angus cow that has very good meat quality, you could have a calf with both desired traits. Another example would be in a population of pigs. If two of those pigs are considerably larger and they are bred together, they could produce offspring that are considerably larger. Okay, so to help put all of this information into more perspective, I would like to discuss porcine reproductive and respiratory syndrome. Historically, PRRS is a disease in swine that is responsible for major loss of economic and biodiversity of species worldwide. Historically, this has been a huge issue. I know I learned about it in college like very early on. Until scientists were able to discover a pig that was naturally resistant to PRRS. This was due to a naturally mutated CD163 receptor in their cells. Now, instead of selective breeding, after scientists figured this out, you can choose to breed the naturally PRRS resistant pig with other breeds to continue the trait, continue passing on the disease resistance. But with genome editing, you can go into the genome of any pig and edit that gene directly at the CD163. So now I would like to invite my associate, Abby Sesker, to talk about an example of genome editing in cattle.
Thank you, Amy. So as we know, the performance of livestock is limited due to many environmental conditions that can cause heat stress and possibly the chance of parasite and disease development. Heat stress can negatively impact milk and meat quality, as well as disturb the effects of animal production and fertility and interrupt homeostasis. In subtropical areas, it is more common to have heat stress. The slick phenotype is a mutation that involves shorter hair with a lower follicle density across the animal coat when compared to the wild type. Senegal cattle were the first one of the breeds that were presented with the slick gene at the University of Florida before it was introduced into Holstein cattle. To dive deeper into this, we recognize that the slick phenotype here, with these cows right there, there is less hair on the face and also on the head and in the ears, which can lead to a generally lower body temperature, which can also result in a greater milk yield, especially when comparing these two summers. There is about a 4 kilogram difference of milk in a day, which indicates that they are much more heat tolerant. Researchers took animals with this slick phenotype to then create this Manhattan plot. Doing this genome-wide associated analysis in this Manhattan plot, it shows the hairy locus on chromosome 3. It was determined that on this chromosome, there was a mutation in the prolactin receptor that was responsible for the hairy phenotype. This displayed what gene could be edited to alter heat tolerance, which was also recently, a year ago, accepted in the US through the FDA. Here, the researchers were also able to see the difference in the tail length between the mutated receptor and the wild type receptor. What comes with gene editing, we have to be worried about what we take with this gene-edited animal or a small population of gene-edited animals. And we want to make sure that we disseminate that genetic information in a new population. This is what is often called the bottleneck challenge. A genetic bottleneck occurs when a population size is reduced to at least one generation of species. Then whichever traits are carried by that one generation are continued as the population increases again. This can cause a loss in genetic variation due to genetic drift, which happens when a newly introduced gene becomes more prominent in the genome, phasing out an original one and decreasing the diversity of the organism's original DNA. This occurs more commonly after bottlenecks and especially in smaller populations, since there is less genetic variation to begin with. The decrease of its genetic variation can cause a decrease in adaptation to new selection pressures like climate change, due to the fact that this gene with that adaptable ability could be one lost in genetic drift. This same problem exists with gene editing to try to avoid this loss of genetic diversity after introducing one gene-edited animal. Now that we are done talking about the technological side of things, I would like to invite Jennifer Holliday to talk about the producer and consumer willingness of gene editing.
Thank you, Abby. Just as Abby mentioned, there are many challenges that need to be taken into consideration when adopting genome editing. Producers and consumers also take into consideration many different factors when they are considering to implement genome editing into their operations or adopt genome editing products into their diets. Looking at the producer side specifically for now, many considerations include efficacy rates. For example, is it successful or is it not? The sale prices, will adopting this genome edit into my livestock operation affect the amount that I can get from the market? Net returns, is the cost that it takes to implement this edit into my operation worthwhile based off of what I can bring back in? Patent licenses, which varies from nation as well as genome editing technique, as well as veterinarian recommendations and peer farmer considerations. On the consumer side, many considerations include price advantages. Is it worth it to me to purchase this cheaper genome edited apple or this more expensive conventionally bred apple based off of what I believe on the following information, including human health concerns, ethical concerns, environmental impacts, and regulations. It is also important to note at this time that much of this information was gathered from studies based off of not only livestock, since it is limited right now, but crops and genetically modified products on the market too. Looking specifically at the producer side, we all believe that sale prices are the top consideration, as if you are not able to bring in a positive income, it will not work out well in the end for your market, which is understandable. That is what many of our studies shown that we looked into, but a second highly considered idea was efficacy rates. A specific study on Johne's disease in dairy cattle was highly considering efficacy rates in its results. For those who may not know, Johne's disease is a disease that affects the small intestine fatally on ruminant animals. In the year of 2007, 68% of dairy cattle operations in the United States were impacted by this disease. In that same year, hundreds of millions of dollars were lost economically internationally due to this disease. So as it can be seen, something needs to be found about it, which is why this study was so important to those scientists in the United States who conducted it. Looking at this efficacy rate chart, you can see that there is a large difference between the 25% and 75% areas. When the increase of efficacy rate raises by 50%, the adoption rate increases by 12% as well. Although 12% may not seem like a lot, The total decrease in economic losses increases by approximately 28%, which equivalates to many millions of dollars for farmers within the United States. Looking forward onto the consumer side of this subject, there are many considerations taken, as we all discussed earlier. It is easily thought of that consumers may go for the cheapest option on the shelf just to save some money, whether you are from a low, middle, or high income nation. However, looking into studies on apples within the United States and France, as well as a GMO study within Nigeria, we were able to find that from this information provided, many concerns worldwide are actually more based on human health concerns, ethical concerns, environmental impacts, and regulations. This information truly is important because if consumers are not willing to purchase their products of genome-edited livestock or crops, then producers would not be willing to produce it as well in the end. So this information must be taken into consideration as you move forward with the future of genome editing to be able to make it economically make sense in the world. Next, I would like to invite Jillian Anderson to come share information on public policy and perceptions.
So we just heard quite a bit about the technical aspects of genome editing, but without policy, none of these technical aspects can continue to move forward. I would also like to note, similar to how Jennifer said, genome editing in animals is relatively new, so most of the information that we have currently is based on plants. A way that many countries regulate their genome edited products is through site-directed nuclease classifications, or SDNs, and there's 3 of them. The first one is small base pair changes or insertions or deletions. SDN2 is small specific changes by homologous recombination of the double-stranded break of the DNA. And SDN3 is precise insertion of DNA elements of foreign origin. In 2018, the European Union Court of Justice ruled that they were going to regulate all 3 classifications the same, even though SDNs 1 and 2 are mutations that could occur naturally, but are just sped up through genome editing. This had impacts on innovations throughout the world. And here you can see how selected countries regulate their SDN classifications. As you can see, the EU is in each of the 3 categories, and New Zealand actually decided to follow suit, so they are in all 3 as well. But you can see certain countries like the United States, Argentina, Colombia, Brazil, Chile, Australia, and Japan only regulate if there is foreign DNA present. Many countries are still in the process of developing their regulations, and developing policy takes time. So examples of countries that are currently developing legislation are Nigeria, Kenya, India, Paraguay, Bangladesh, the Philippines, and Indonesia. And as of right now, they typically regulate products on a case-by-case basis as they come up. Something that shapes policy is public perception, and policymakers must consider public opinion if there will ever be synergy between the science and the public. During our research, I read a study on a focus group that was done in the UK of 38 participants. They gave these participants information on genomic technologies, which doesn't just include genome editing, but it was one of the 6 technologies that they discussed. They gave participants information on the technologies, asked them their concerns and their excitements with this, and then they had a large discussion. Some of the points brought up by the participants were that using these technologies for conservation of ecosystems and species was important to them. Another thing of importance was using it for animal welfare and disease resistance, and they were curious on how it would impact their food systems. They had some ethical concerns with the technologies, and there was also a fear of motivation by profit alone. Something that was not mentioned in the study is that while it was small, it reflects public sentiment as a whole. Something that wasn't brought up was confusion with terminology and how that can impact public perception. As you can see, this is a very complex topic and there's lots of terminology that goes on with it, but neither science nor human innovation fit neatly into a box, which can make it hard to have concrete definitions with the science. Another consideration is patents. As CRISPR technology specifically develops very quickly, there's more and more patents coming up and also more and more stakeholders coming into the game. That can be a good thing or a bad thing depending how you look at it, but one of the risks is exploitation of low and middle income countries and their genetic resources. One way to safeguard against this is through access and benefit sharing. The main way is through the Convention on Biological Diversity, specifically their Nagoya Protocol. What this does is makes it so that there has to be mutually agreed terms and prior informed consent with the countries that have the genetic resources of origin, as well as the countries who would like to use it. This ensures that there is not exploitation of low and middle income countries. Something that's come up in recent years, however, is digital sequencing information. Because technology has developed so much, you no longer need physical DNA in front of you in order to know its sequence, and a lot of this is stored in databases. So the question becomes, does the Convention on Biological Diversity cover DSI? This is a divisive topic, and there's even some legal questions with it, as let's say an institution has access to a database. Do they have a legal obligation to share their benefits with the country of origin that that DNA came from? There is an urgent need for a conclusion to be met on this subject, because whether or not the Convention on Biological Diversity accepts DSI or rejects it, there will still be major ramifications for the future of genome editing. Now that we've talked a little bit about the policy, I would like to invite Amy to talk about the future of this technology.
Thank you, Jillian. Part of what drew me to this project in the first place was what the future of genome editing can do for the field of animal science, especially when it pertains to the protection of genetic diversity. As we know, disease can cause the death of entire populations of animals, which in turn will cause a huge loss of biodiversity. Now, I've already talked about PRRS, but I would like to highlight another very relevant disease to swine currently. It's the African swine fever. I chose this graph from the World Operation for Animal Health because it shows a number of outbreaks, cases, and losses within only the weeks of March 31st to April 13th. This is how much this impact has had in only 2 weeks, the disease on populations of swine around the world. So the question remains, can we use genome editing to combat African swine fever in the same way we did PRRS? Well, not we, but scientists. So what do we know? We know that we have the technology to identify the gene within the genome, as we did with PRRS. It was the CD163 receptor. What we don't know in African swine fever is the target. But what we do know is that we have the technology and the scientists to get it done once we figure out what the target is. So who's to say we can't combat other diseases in other animals in the same fashion, and thus helping prevent the loss of biodiversity of every species that has to deal with disease? Thank you. I'd like to invite Jillian Anderson back up to finish out our presentation.
So another question that comes with the future of genome editing is whether or not it can be used for both native and local breeds and commercial breeds at the same time. So historically, genome editing has been easier to do in commercial breeds because that's where all the research has been and that's where the resources typically are. And there could be cultural considerations to go along with this as well, but one of the bigger issues is dissemination. So once a producer in low and middle income country has a genome edited animal, how do you get that out to the rest of your population or your herd? Once you have a genome edited animal, it requires an intense genotypic and phenotypic analysis in order to ensure that the gene edit did what you wanted it to do. And a lot of times in these countries, there's a lack of infrastructure for that long-term phenotypic analysis. So the issue of dissemination is a big question remaining. So we've done a lot of research on this topic, and through this, we've come to our own conclusions. We believe that genome editing is a tool which has the potential to aid producers in low and middle income countries combat the effects of climate change on their livestock if the following happen. Equitable policy needs to be established. Biodiversity must be maintained. Balanced information needs to be provided to producers and consumers. And technical barriers need to be addressed, like the bottleneck challenge and understanding phenotypes so that we can define potential genomic targets. For the outlook of genome editing, climate change is going to continue to pose problems for livestock producers, particularly in low and middle income countries. Policy and public opinion are going to continue to ever evolve with the science, as well as with the cultural, social, religious, and moral considerations that go along with it. And we believe that the use of phenotyping technologies will be crucial for genome editing in livestock moving forward, and they must be used together. With that, we would like to thank our FIO colleagues for all their hard work they did with us, and we are now ready for any questions you have.
So the preference for those online is that we take the questions from the people online using the chat feature rather than the live feature due to potential audio problems and so forth. So I guess we do already have one question, and this is from Axa Villalobos. And his question is, is genome editing need to be considered under the Cartagena protocol? Have any of you looked at that aspect?
So I did more research on the Nagoya protocol than the Cartagena protocol, but I do believe it is that genome editing must be considered under both, because those are live animals, especially in the Cartagena protocol that deals with live animals. If genome edited animals become more prevalent, they will definitely have to be considered under that agreement.
Okay, thank you very much. I hope that addressed your question, Alex. So if there are any other questions, I'll just inform you that we will have time for overall discussion later, so there's no urgency. But if you have any other questions, we could take one or two right now. So there is one more, or we have two questions. I'll take these two and then we'll hold off for the others later. So the first question is, how can we make genome editing techniques affordable in developing countries?
That is a great question that we are all honestly still trying to figure this out. Honestly, a great example of trying to decipher this is in Africa, where we have found through talking with Dr. Mike McGrew that there is much freedom within patenting there. A lot of the challenges we have discovered within making it affordable comes down to the patents and having larger companies and corporations take over the genome edit technique. So it must be asked, or they must pay to be able to use it in the future. So by finding ways to make it more affordable through patents, or not allowing patents in that way, but that's also a hard way to balance it, because also the amount of money it takes to develop it must be brought back in the end for those investing. So it's a hard balance that we are still trying to move forward with.
Okay, then we'll go, just one more question. It's from Pablo Serra. First, he wants to congratulate and thank you for your hard work. His question is, have you considered animal ethics concern in your study?
Yes, so in a lot of my readings, as I focused more on public policy and perception, there were a lot of ethical questions brought up by participants, for example, in that focus group that I was talking about, but also in other papers I read as well. It really comes down to individual personal morals, as well as what governments will allow with animals. So yes, ethical concerns are a big component.
Now, I'll also add, in our discussion with our ethics expert,
yes,
she also pointed out that it's really a case-by-case basis that
yes, and
you'll have
there's
to look at.
A lot of people are concerned that genome editing isn't fixing the root problems, so that's another ethical concern as well.
Well, thank you very much. So right now, as I mentioned, there are some more questions, but we'll save the rest for the discussion period, and we'll move on to our second presentation on phenotyping with relationship to climate change, and also considering low and middle income countries.
Good morning. Sorry. Good morning. My name is Rebecca Larson. This is Molly Simmons and Shraddha Salori. and we will be talking about the challenges in livestock phenotyping. Now, as the other group mentioned, there is a need of an analysis of genotyping as well as phenotyping, which is required to have the best outcome for producers of low and middle income countries. And our objective of this presentation is to review traits and phenotypes of the big 5 livestock species, which include cattle, sheep, swine, goats, and poultry, relevant to global climate change. And we will discuss phenotyping technologies and challenges of implementation. And then our methods. In the paper, as well as our presentation, we conducted extensive research by reading and annotating scientific publications. And from the help of Paul Betcher, our FAO colleagues, and our professors, we were allowed to interview with experts in their field, including Dr. Rosa, Dr. Marode, Dr. Deckers, Dr. Seibel, and Mr. Blythe Schultz. And then the bigger picture, so this slide deck presentation is based off of a bigger piece that our team has written. Our paper is based off phenotypes in low and middle income countries in relation to climate change, the Sustainable Development Goals, and the big 5 livestock species. And then dealing with our overview of what we're going to be covering, we will go over the global importance of livestock, the impacts of climate change on livestock, defining phenotyping, phenotyping for heat tolerance, phenotyping technologies, challenges of implementation, and then we will discuss the opportunities. And then I'll hand it off to Shraddha.
Thank you so much, Rebecca. I will be going into a little bit more detail on the global importance of livestock. As we all know, livestock plays a crucial role in addressing key challenges related to sustainability. According to Shaping the Future of Livestock, report by FAO, from the perspective of sustainability of the global livestock sector, there are 4 important and interrelated aspects, including food and nutrition security, livelihoods and economic growth, animal welfare and health in relation to human health, and also climate and natural resources. In terms of food and nutrition security, as the world population increases, there is an increasing need of animal source food, especially in the sub-Saharan African region and South Asian region of the world. And animal source foods such as eggs, milk, and meat are highly nutritious and are needed to ensure nutrition security around the world. However, there is still disparity in the distribution of these animal source food products in low and middle income countries and high income countries. Moving on to the livelihoods and economic growth aspect, livestock is also an important asset in sustaining livelihoods, especially in the LMICs. They act as a safety net and help alleviate people out of poverty and also help build resilience. Animal health and welfare is closely linked to human health. Zoonotic diseases are affecting human health, and they have the potential of becoming global pandemics, as we have seen in the past. Therefore, it is essential to have good animal health and welfare practices in place to prevent the spread of such diseases. In terms of natural resources and climate, livestock draws heavily from natural resources like land and water, but they also have the potential to contribute to the conservation of biodiversity and genetic resources for food and agriculture. And if managed sustainably, livestock can contribute to important ecosystem services and maintain agricultural landscapes. All of these 4 aspects are derived from or are based upon the Sustainable Development Goals of the United Nations. Next, I will be moving on to the impacts of climate change on livestock. The ongoing climate crisis is putting immense pressure on the livestock sector and is affecting the production and efficiency of animals. Some examples of climate change include, like the effects of climate change include rising temperatures, increased spread of disease, feed quality and quantity, and water scarcity. This in turn is having impacts on other issues related to reduced growth in animals, reduced milk production, reduced reproduction, and meat production. In order to address such issues, phenotyping must be used to improve production efficiency, genetic advancements and potential, management practices, and overall sustainability. Just to set the scene up a little bit more, I would like to provide two key definitions for this presentation. The first one is phenotype, which is an observable characteristic or trait of an organism. And the next definition is phenotyping, which is a technique used for measuring the phenotype of pre-selected traits in specific individuals. The genetics of an animal and the environment it is brought up in determines the phenotype of that particular animal. Now I'll be talking a little bit more about phenotyping in heat tolerance specifically. As climate change worsens, there is a need to monitor adaptive phenotypes like heat tolerance and how we can adopt them into our breeding programs. Here are some traits to show direct response to heat tolerance, to heat stress, excuse me. And these traits include respiration rate, rectal temperature, and skin and surface temperature. Next are some examples of traits that are correlated to heat stress and can act as indicators to tell whether an animal is under heat stress or not. And these traits include body size, hair coat, skin color, feather density, and hair density. Lastly, these are some traits measured to quantify the response of an animal to heat stress. Heat stress and all of these traits mentioned on the right column show that there is a cause and effect relationship, which means that there will be a change in these observed traits if an animal is exposed to heat stress. In this presentation, we will be particularly talking about hair coat, hair density, feed intake, growth rate, milk production, and feed efficiency. Now I'll be moving on to talking about heat stress specifically in swine. So pigs are impacted by heat stress because there is a deficiency in the functional sweat glands, and that help them facilitate their body temperature and heat loss by evaporation. And they also have a thick layer of subcutaneous adipose tissue that acts as a layer of insulation. And these are, pigs are also homeothermic animals, which means they can maintain their core body temperature only within certain limits. And if they transcend those limits, they tend to experience heat stress due to increase in ambient temperatures. And they tend to show signs of increased respiration rate, decreased feed intake, which also has impact on decreased average daily gain, a decrease in feed efficiency, and their overall performance. These are the traits that were focused on by Rao et al. In their study, and I will be giving a little bit more information about the study itself and the results that were presented in the study. So the study focused on the effects of repeated heat stress on pigs and measured the growth performance, feed intake, and feed efficiency in commercial pigs. To summarize, the results of the study showed that there was a sharp decline in both feed intake and feed efficiency when the pigs were constantly exposed to heat stress periods. The reduction in feed intake is a coping mechanism used across all the 5 livestock species to reduce their internal heat production. Reduced feed intake also has impacts on other metabolic functions, as mentioned previously, like reduced growth rate. Moving on to some challenges in measuring feed intake and feed efficiency as traits. Even though feed intake is a strong response to understanding whether an animal is under heat stress or not, it is oftentimes a huge challenge to measure this specific trait. Challenges can come in terms of systems, so intensive versus extensive systems. In terms of intensive systems, these systems have huge populations of animals in confined spaces, and they are fed in groups sometimes, so it can be difficult to measure how much feed is being consumed by individual animal. The same sort of logic applies to extensive systems as well. They are more pasture-based systems, and animals are usually free grazing. which makes it difficult for farmers to keep track of how much feed is being consumed by each animal. Other kind of challenges include measuring feed intake and efficiency being time-consuming and labor-intensive. However, there are still opportunities in terms of applying or implementing phenotypic technologies that my colleague Molly will be talking more about. But in this case, to measure feed intake, there are electronic feeders that can monitor how much feed is being eaten by animals. And in low to middle income countries where there are smallholder farmers who have smaller number of animals in their farms, they can try to monitor the feed intake of their individual animals. Now I'll be giving the stage to Rebecca, who will be talking more about coat characteristics in goats and sheep.
So coat characteristics, there are different signs for this, which is color, length, and density. And then for goats, within breeds of goats themselves, white goats are more heat tolerant than any other color, and because white coats reflect heat better than any other color. So this can lead us to say that white goats are more tolerant to hot and humid conditions and can be implemented anywhere around the world that has the hot and humid conditions, but there are trade-offs to this. For goats that have white coats, in studies, it was shown that there was lower fertility, lower reproductive performance, higher feed intake, as well as lower water consumption compared to all other coat colors. And then moving on to sheep. Sheep with hair are more heat tolerant than sheep with wool, and sheep with short hair are more tolerant than sheep with longer hair. And according to studies, light-haired color is more tolerant than dark-haired color. So this can lead us to believe that short-haired sheep with lighter hair color are more tolerant in hot and humid conditions and are better implemented all around the world. But there are challenges to measuring coat characteristics because coat is correlated to heat tolerance, so it is not easy to measure as a standalone trait because it's subjective and there's no standardized measurement for coat characteristics by themselves. And then I will hand it off to Molly.
So another important phenotype to measure is milk production. As Shraddha mentioned earlier, milk holds very high nutritional value for people all around the world. However, we see that there's a negative correlation with rising temperatures and milk production yield. With a long-term exposure to high temperature and humidity, dairy animals experience increased respiration and dissipation of heat in order to maintain stable body temperature. However, long-term exposure to high temperature and humidity can compromise this function and lead to decreased growth hormone in the dairy animal's body. This ultimately leads to decreased production. And we have been able to track this phenomenon because milk production is one of the most widely common measurable phenotypes in the world. But there are a lot of measurement challenges that persist in improving our knowledge of milk production. One of those is accuracy. If milk yield measurements are not accurate or not taken on a daily basis in the long term, then further research regarding the relationship with temperature and milk production will be compromised. Additionally, there is a lack of independent organizations with established record-keeping systems, especially in low to middle income countries, that could facilitate in long-term milk production record-keeping. We've just discussed some traits that can be related to heat tolerance, but now we're going to move forward to talk about the types of phenotyping technologies we can use to measure these phenotypes. Today, we're going to categorize some examples into high-tech interventions and low-tech interventions. In high-tech phenotyping technologies, we see a lot of precision livestock farming. That is the use of sensors and actuators to monitor physiological response or behavior traits in animals using real-time monitoring. Examples of this could be milk monitoring systems, where every milking period, individual animal is tested for somatic cell scores or for milk quality. On-animal sensors can be used to see and show signs, demonstrate signs of symptoms of lameness, or can be a way to measure physiological conditions in an individual animal. Image-based technologies are growing in popularity, especially in intensive systems or in places where there's not enough labor to help on the farm. These image-based technologies are real-time images, real-time monitoring of animals, and it can help measure behavioral responses or show some early symptoms of disease and lameness. Like I kind of alluded to, a lot of these technologies are used in capital-intensive systems, and a lot of these systems are found in high-income countries. These high-tech technologies are mainly used in those systems because of economies of scale. They're expensive, and if it's not economically feasible for a farmer to use them, then they won't use them. But low-tech interventions for phenotyping technologies are used all around the world, regardless of high, low, or middle-income countries. These could be simple digital tools that allow for a place to record phenotypes or to get feedback from a veterinarian about the performance of your animal. Calibrated buckets could be used to measure daily milk yield if an intensive type monitoring system isn't accessible. And then measuring tape for body size to estimate body size is commonly used around the world. And finally, growing in popularity is the use of mobile apps to record performance data and to, again, help facilitate in a two-way communication feedback. However, with all of these technologies available, there's still a shortage in phenotyping around the world. And that's because we have a lot of challenges of implementation in, especially in low to middle income countries, but also in high income countries too. When we're thinking about implementing phenotyping technologies or developing programs to implement these technologies, we need to consider these factors and the cultural. I'll talk about the cultural and social factors first. Factors to consider are gender. We need to make sure that we are not missing out on an entire group of people that could be making a positive impact in the development and use of phenotyping technologies. Also, we need to match the type of technology we are implementing with the education level of the farmer and the community that we are giving these technologies to. And then, of course, we need to consider the cultural nuances. Not every solution is a one-size-fits-all. Everybody has different belief systems that will impact how they view the phenotyping technologies. In the capacity section, it's really important to note that infrastructure is a big barrier in adopting more phenotyping technologies. If people don't even have the simple tools to measure phenotypes, like a measuring tape, for example, they're not going to develop or utilize maybe high-tech technologies. It won't be accessible. Also, it's important to consider access to electricity, data storage, and other factors regarding infrastructure. And then technical knowledge, if somebody doesn't have access to maybe data specialists or livestock specialists, they may not be able to troubleshoot issues with the phenotyping technologies they have. Governance is a huge issue. We need to, when we're developing these phenotyping technologies and the programs to disseminate them, it needs to incorporate the theory of change, looking at these issues from a systemic approach and making sure that what we're doing is more locally led so that it can be sustainable. And then kind of coming along with that, it's important to consider the livestock system that we're talking about with phenotyping technologies. Extensive systems cannot use the same phenotyping technologies as intensive systems and vice versa. Finally, it's important to talk about the economic factors in implementation. Like I talked about earlier, if implementing a phenotyping technology is too expensive for the farmer, they're not going to use it and it's not going to be feasible for them. Another issue with phenotyping technologies and their dissemination is a lack of private investment to help fund programs to get hit the ground running for these technologies. And then finally, it's important to consider whether the technologies we're giving to farmers are going to take a lot more time, because if it requires a lot of time for them to record phenotypes and use these measurements, then they might not use them. So there's a lot of barriers surrounding phenotyping technologies, but there's also successes and opportunities for implementing phenotyping technologies, especially in low to middle income countries. An example of this is the iCow system. It's a digital tool offered in Kenya, Tanzania, Ethiopia, and Nigeria. Farmers can use this free service on their phones, mobile phones and cellular phones. They can receive SMS messages regarding management tips, along with huge access to a library surrounding the topics of feeding, disease control, and milking assistance. So these types of information and dissemination information can help them improve their production. There are also audiovisual elements for people that can't read. If they have a mobile device, they can access a library with audiovisual elements to help them improve their management and production. A really important feature of iCow, though, is the data capture tools they have. So it's a simple digital tool with manual entry. Farmers are able to put in body scoring measurements, milk production, and calving dates, so they can keep a long-term record of this. And then with the service, they can also talk to a veterinarian if they need to, and utilize these performance records to help facilitate with questions for the veterinarian. A fruit of this software and program has been contributed, is contributing to the African Dairy Genetic Gains Program. This program was established in 2016 with collaboration of the International Livestock Research Institute and the Bill and Melinda Gates Foundation. It was started in Tanzania and Ethiopia, but has since, from success, expanded to other African countries and is starting to expand to some Asian countries as well. With collaboration of iCow, they can take on-farm performance data with the farmers that have consented to do so, and take that data and give actionable feedback to the farmer. If a livestock specialist seeing the data sees like an issue, maybe an extrinsic factor has lowered their milk yield, the livestock specialist can relay that back to the farmer to help. But they also can take samples from the farmers with their permission to conduct genotypic and phenotypic analysis. This has helped facilitate a breeding program in which they can use the information to select crossbred bulls for genetic improvement. These two examples are great examples of how, with demonstrated positive impacts of short-term phenotyping, farmers see the impacts from improved management and production. It encourages them to establish long-term performance recording, which can create genetic improvement programs like the African Dairy Genetic Gains Program. A big takeaway from all of this is that we need to give farmers positive, when we're implementing solutions, they need to see the short-term impacts so that we can improve in the long-term genetic advancements and help them adapt to climate change. And with that note, I will hand it over to Rebecca for conclusions.
In conclusion, measuring phenotypes can improve management and production of livestock, and long-term performance recording can contribute to genetic advancements as well as contribute to breeding programs. And because climate change is affecting production, phenotypes are critical to measure heat tolerance, and we need to be able to measure direct, correlated, and quantifiable responses. And although there is technology available, there are many challenges existing in the implementation of phenotyping. We characterized these challenges earlier into 3 categories: cultural and social, as well as capacity and economic challenges. In the end, producers need short-term impacts to continue long-term performance and management of recording phenotypes, which will create better opportunities for genetic advancement. I want to thank everyone who came today, as well as our amazing FAO colleagues who include Paul, Christian, Gregoire, Roswitha, as well as Rama, and our wonderful professors, Carmen, Stephen, Shelley, Elizabeth, and Joe Coletti. Thank you again, and we will move on to questions.
Okay, we'll start with a few questions on the phenotyping aspect, then we'll open it up to wider questions. Okay. So we have a question from Sandeep Banerjee from India. So he wishes to greet you from his institute in Calcutta. He's saying that the day temperatures now is 95 degrees Fahrenheit, which is I guess around 36 Celsius, and 75% relative humidity. The frizzled plumage, naked net conditions in chickens are adaptations in hot and humid regions. There are similar traits in small ruminants, cattle, buffaloes, camelids, and other species. However, the overall direct economic output of such unique breeds is much lower than commercial breeds. How can we balance these, these aspects, the balance between adaptation to a given climate and productivity? So many times these unique livestock breeds are lost, especially when marketing their products are not regulated relative to the commercial breeds. Did you find any discussion on that or draw any opinions based on your research?
So what I gathered from that is that comparing naked neck and frizzled chickens to regular production birds, like why aren't they implemented more? When I was looking into actually both of those breeds, it's actually mentioned in our much bigger paper about phenotyping specifically that, especially for the naked necks, for those of you who don't know, it's a chicken who doesn't have any feathers on its neck. This helps with them regulate heat, keep them cooler. They're more adaptive to hot and humid climates. But a downfall of implementing these and why they're not implemented more worldwide is because one of the challenges that I found in my research was that they're not accepted. They're not in a lot of areas indigenous, and people don't want to implement them because of their looks, and they don't think that they'll be as as tasty, or they will have such improvements as the indigenous birds or the birds that they already have. So I think it's that lack of knowledge aspect, as well as that cultural, as well as the unwillingness to implement them.
But if they're breeds that these features within their own native area, how could it be a policy issues could help implement breeding programs? Could that be part of it rather than policies that may favor commercial breeds? Have you found any evidence of that as well?
I was just going to mention. So I'll just mention something really quick. So when I was looking into frizzles, even within the United States, they're a more expensive bird. So even if they are more heat tolerant, it has to do with that economic aspect and the price difference, that there might be other chickens especially that might be well adapted, not the best adapted for heat, that can suffice for the farmer's needs. But I'll pass it.
I think with that, it's really important to consider the cultural aspects. And I lost my train of thought. Give me a second. Can you repeat your question? Sorry.
No, I just mentioned that the question was generally why are reasons like adapted animals within local climates maybe sometimes fail against
so our project was about phenotyping, and I think one of the big issues with this is we have a lot of phenotyping data on commercial breeds because of long-term performance recording, but there
isn't necessarily a lot of recording on certain indigenous breeds, which is also why we have issues with genetic diversity in livestock, because we don't, there's still a lot of livestock species, indigenous livestock species that haven't been recorded or reported. So it's really important to get that first step going and to acknowledge the importance of these indigenous breeds and to work towards establishing their genome and establishing knowledge of their phenome so that we can work towards improved dissemination and knowledge of these breeds.
Was there any question from the floor based on phenotyping aspect? Then we'll move it to the overall discussion again.
Thank you. Xiang Yusong from FAO Leap Partnership. I have a question based on your presentation. I know the phenotyping or measurement is important to the farmers, but could you elaborate a little bit? What's the motivation for the farmers to measure this? What's their short-term benefit? How you encourage or motivate them to do so? I mean, I understand it's labor-intensive and farm work is hard. So in a small-scale farm or developing country, it's hard for them to do so. And on the other hand, for the developed country, the technology is expensive. So what's the short-term benefit for them? Thank you.
So within our research, I really focused on milk production, and I think that's a great example of the short-term impact through daily monitoring of milk yield. When a farmer sees a drop in production, that could be an indicator to them that there's this extrinsic factor going on, a disease, or maybe it's a response to heat stress, so that they can measure it and keep note of it and understand and hopefully treat it. Right? That's what we want to do with phenotypes, is to record them so that we can be proactive with these extrinsic factors.
So I'll go back to one more question from the chat. This is from Maria Wurzinger, who's from Austria, uniquely, considering the room we're in. And she once again, thank you for the nice presentation. And you had mentioned that there is often not enough investment from the private sector in phenotyping activities. What role could the government institutions have, either to, jump-starting phenotyping, or is there maybe some sort of, common good that they could justify investment? So, what do you see about the role of government in phenotyping?
I think where we're seeing a lack of private investment is especially in low to middle income countries. And this can be a little tricky because government institutions may not always have the capacity or the governance to do so. So that's why I mentioned private investment, because that's where a lot of it is coming from already, like the Bill and Melinda Gates Foundation, for example. So I think it really just takes a team effort, and it's really important that if government institutions are going to fund it, that it has good governance so that these programs can be sustainable in the long term.
Okay, so what I'll do now is I'll go back to some of the chat questions which had been on genome editing, and then hopefully we'll have more questions on both topics as we go on and we can continue. But there were some more from the genome editing aspect. So one question related to genome editing, what consideration do we have to have in mind to the fact that genes often influence more than one trait? So in theory, if you edit one trait, there could be effect on others. Did you find any discussion of that, or do you have a developed opinion on what could be done to address those potential issues?
Yeah, I can take this one. So obviously, we've been talking about monitoring and how important it is to talk about, in comparison, phenotyping and genotyping. When you change the genome, you hope to know what's going to happen and you hope to have more control, and that's why genome editing is so important, because it's more precise, especially with the old practices of selective breeding. That's why I wanted to talking about it. People are going to try to figure out new innovative ways of agriculture, so genome editing is a more precise way to do this. Obviously, there's always traits that you aren't going to consider, especially when I was talking about the African swine flu. We know that the CD163 receptor exists in PRRS. What I didn't say was there could be one receptor in African swine fever, there could be 10, but what we have is the technology to figure that out. So obviously there's going to be factors that we don't know, but the important thing is we have the technology to figure it out. Hope that addresses the question.
Was there any policy issues about that? Or ethical issues?
In my ethics research, off-target effects were definitely something that people were concerned about, but as far as the science goes, there isn't that many more off-target effects with gene editing than there is with traditional breeding. You have off-target effects with traditional breeding as well. So I think that's kind of where the education aspect comes in as well.
And there were several kind of related questions, so I'll try to group them into one. Could you reflect a bit more about the access to the technology and the knowledge needed for genome editing, maybe how that differs around the world and so forth, and the barriers that are going to be important if they differ in different parts of the world?
Yes, so as I mentioned earlier in the question that I answered about patents, patents really do influence the availability based on the nation that you are in, as well as the economic barriers. When we are looking at the United States and basing information off of genetically modified organisms, we can see the development as they have been around for many, many years and in the market. So we can see the development based off of the 10 GMO crops that are available in the United States, or in the EU, no cultivation of GMO crops is allowed. And it really is a lot based on not only economics and the technology availability, but oftentimes it comes down to policy impacts as well.
And also, CRISPR is something that is pretty accessible. It is a pretty quick and easy way of gene editing, and it is being used even in high school classrooms, so I'm sure it can easily be shipped to middle and low income countries to be used there as well.
So this links into another specific question. Did you, in your research, find examples of this CRISPR being used in laboratories in low and middle income countries? I mean, even maybe in other plants or livestock, or even other sectors of the agriculture economy?
Yes, there was research found, but I'm not exactly sure on low and middle income countries. I know it's being used in certain areas, but not everywhere that it's needed.
The difficult part with this technology is that it is expensive and it's difficult to use. You have to have the right experience and you have to know what you're doing, because what you're doing is so technical. So it's the toolkits for CRISPR are made to be easier to use, but because the process itself is complicated, it's a little bit more difficult.
So you require international cooperation, I assume,
Yeah.
to really get it off the ground?
Yeah, it depends on how much technical knowledge is available in those areas, because CRISPR itself, compared to other genome editing technologies like TALENs or ZFNs, it's cheaper and less complicated. So it really just depends on the level of technical knowledge that exists in those low and middle income countries. It is being used there, but to a lower degree than in high income countries.
So now I'll go back to a more phenotyping question. So this question specifically, it's from Kwenga Namge from Bhutan. You mentioned that the wool sheep are less heat tolerant than the hair sheep. Is there anything that can be done for wool sheep? I think both from a management perspective or breeding, if you encountered anything with that and so forth?
So in terms of making them more heat tolerant, okay. Oh, there you go. Sorry about that. So in terms of making them like less heat stressed or a little more heat tolerant, there's shearing that you can do on wooled sheeps that can make them a little less stressed due to heat. And yeah, so those are that main practices in terms of like shearing. And in the research that I conducted specifically for sheep, it did show that the results showed that shearing helped them release their sort of body temperature and made them a little less heat stressed. So that's a practice that can be done.
We have questions or comments from the
I have a more general, I mean, it goes a little bit to both teams. And first of all, many thanks for these wonderful presentations and the great work you did. That's number one. So one of the biggest threats for the erosion of genetic diversity is what we know from many countries is indiscriminate crossbreeding. This means bringing exotic breeds to other countries, quite often developing countries, and then somehow with artificial insemination, distributing in a very uncontrolled way the genes. How do you think with these technologies you're mentioning, this will increase the threat of losing diversity and having more and more indiscriminate crossbreeding and bringing exotic breeds to these countries?
I feel like when it comes to this topic, it can be kind of tricky on how we think the animals will adapt to the environments, and that's something that we really need to consider. But when it comes to the technologies, it's kind of a gamble that you take, but it's also really scary to think that we could lose all the genetic diversity of these exotic animals. And it's just something that we really need to keep in mind and make sure that with the bottleneck challenge, we don't lose those factors of the exotic animals.
I also briefly mentioned cultural considerations.
It's not our wonderful Holstein cattle that are under threat. It's the other way around. So it's not that the local adapted native breeds, we are going to lose them maybe.
I briefly mentioned cultural considerations, and that's an important thing to incorporate into policy specifically. With indiscriminate crossbreeding, if there's no policy to prevent it, people are going to continue to do it. So there needs to be policy with cultural and indigenous voices included. I did some research and I read a paper about the Maori population and their views on genome editing. And something that they were worried about was losing their native species to these technologies. So cultural considerations must be taken into policy.
In addition to that, part of why I covered disease is because disease is a loss of genetic diversity. So with genome editing and in relation to phenotyping, we can sort of use all of our knowledge to be able to combat the loss of biodiversity in other ways. So it's not directly it's not directly addressing the problem, but we can help and aid other issues that are affecting it.
Okay, I guess go ahead.
I've got actually a technical question regarding adoption of genetic edited animals. Would you suggest, or did you find some kind of metrics to measure in what extent there is a development of genetic editing? I don't know, for instance, authorized patent, population, or something like this. Would you have some suggestion, or did you find something about that?
I'm sorry, could you clarify your question a little bit more?
How do you measure the extent of gene editing in livestock?
So with regulation, as I mentioned, those SDNs, each level is a different level of how much that animal was edited. So that would be one way to determine how much editing has gone into that. As far as patents go, patents are individual by country. So if you have a patent on something in one country, you don't have that same patent in another country. So I think it's important to use a standardized measurement for how much editing has gone in and use that to build your patents, if that answers your question.
Can you measure how many animals in the world are gene editing?
It depends on if it's reported or not. I think that's where policy comes in. I mean, it all comes back to policy for me, at least.
You also have to keep in mind what's being done in labs is a lot more extensive than what's being done in the real world so far, because we just need to wait for it to be accepted and how the public is going to take it.
I'll take one more question from the floor.
Hi, everyone. My name is Sarin. I'm an intern with NSAG, and I'm a veterinarian. I just graduated. First, congratulations to all of you. This is amazing. I wish my university had a similar program. This is amazing. And yeah, my question is, we have talked about a lot of issues that we are going to face on a producer level and a consumer level. Another issue that I think you might have missed is the misinformation out there, because I come from India, and even today I read a Facebook post about how broiler chicken are full of hormones and how they're keeping us unhealthy, that kind of stuff, which is not backed by the science, and veterinarians have to explain to the public. And so this is the spread of misinformation. This is going to be huge when it comes to gene editing, apart from the original ethical concerns or environmental impacts it might have. But how are we, or how you are suggesting that we manage this misinformation epidemic?
I think that is an absolutely amazing thing to point out. I 100% agree with you. And within conducting my literature reviews, a lot of the studies that were conducted were based on providing the sample, the people that were in the study, on correct information about what genome editing is, genetic modification is, and conventional breeding is, and teaching them the difference between the three. Because then eventually, as the education continued within these studies, the rates of acceptance changed within the sample audience. So I agree with you. I think it's something that truly needs to be addressed around the whole world, no matter what country you are from. It really will come down to, I believe we have discussed farmers and the occupation of producers willing to educate those that are questioning it, as well as education systems and public policy. Public policy and banning things can create fear in consumers. And if you are creating fear, that leads to more miseducation too.
Another thing I mentioned briefly was confusion with terminology. I think it's important for the people who do know about these technologies to use consistent and correct terminology when referring to it, because if we don't know a consistent way to talk about it, how are they supposed to know? So I think it's really important to be consistent and precise with your terminology as well.
Sorry, I would just like to quick shout out Rama for, she's in communications at FAO, and she talked to us a lot about a social media aspect, and it put sharing all of the information that we have researched over months into perspective. It's really hard to take a topic like this and explain it effectively. And the thing that you always have to consider is, what is the message that you want to send? And once you figure that out, everything else isn't easy, but it's doable.
So I'll just kind of close this session by asking one question that really could be answered by anyone in the group. So you can even take turns if you wish. You've studied there that I believe that you found there's a potential for both new and existing phenotyping and genome editing to help, in theory, the developing low and middle income countries both achieve their food and nutrition security goals and preserving their local breeds or animal genetic resource diversity in general. Do you have a feeling that that these technologies will be used in this way, or are you a bit skeptical? And if you're skeptical, what could be done to help ensure this goal rather than not reaching the goals? So anyone who wants to go can give their viewpoint on it.
I'll touch on phenotyping, and from personal experience and my personal evaluation of something that we talked about. It's super hard to measure feed efficiency, and I actually worked for a U.S. organization that was doing feed efficiency, and it takes a lot of work, and that's like the like current. They don't want to keep doing something because they can't see the long-term effects like in the short term. And for trying to measure feed efficiency, you had to take all of the feed out of a bin. Like it was specialized for cattle, so like every cattle had their own like eating box. Then you would have to take it out and weigh it every single morning, every single night, and it gets it's very time intensive, labor intensive. And so it's really getting the education across that, you know, it might be very time consuming and it might take a lot of work, but in the long term, doing the short term work can really lead to long term impacts. And, you know, in these areas, they call them, you know, like cash cows, you know, in low income countries, low and middle income countries, that, you know, this is something that can really improve them if they're struggling to do a little work now for really good impact in the future, just by trying to measure, you know, the feed and putting in a little labor for it.
Any other opinions on this big question of whether these technologies will help and what needs to be done to, you know, to improve their utilization or
Yes.
Yes.
So, excuse me. So coming from my standpoint, I think a great study that I discovered was on a lower middle income country in sub-Saharan Africa. So based in this study in 2012, this country's average household income was $3,000 US. And then the study continued on to share that by the year 2050, if low and middle income countries don't continue to adopt genome editing techniques, they will lose $1.5 trillion. That is a lot of money. When I did the math, 3,000 fit into that number 500 million times. That's 500 million families who could be saving money and finding other ways to continue to feed their family and reduce food insecurity in their households. So I truly believe that just based off of this information, which also a statistic that I think is important before I continue on, was 34% of those polled within this survey were still not willing to adopt genome editing products into their diets. It needs to be considered that, as you mentioned earlier, education needs to be a large factor of this all. Consumers need to be willing to adopt it into their diets, and the governments of these countries need to begin to find ways to incorporate it and allow their communities and those that they're representing to consume them, to decrease food insecurity, because that truly is, I think, one of the most prominent issues that we need to be focused on.
Quickly, I can say it again, policy. Policy, especially in low and middle income countries on these topics, are still being developed, and it takes a lot of time. So personally, I foresee these technologies being used in the future, and I think there can be benefits if the issues we talked about are addressed and are addressed equitably. But I think it'll just be a matter of time until the policy can develop and international agreements can be reached.
I would like to say that personally, my passions lie within ecology. And I would like to look to see, after this, I'm going to do a lot of research on how genome editing can be done to improving coral reefs in the ocean and improving wildlife species, just like Roswitha has been saying. And I feel like there's a lot of potential for genome editing outside of agriculture as well. So even if it's not being considered by producers, it should be considered by the rest of the world in other aspects.
I would say with both of the issues of genome editing and phenotyping technologies, there is a need for infrastructure and better governance. That is a big barrier for both of these issues. In the context of climate change, we really need to scale up investments to improve livestock and help livestock adapt to climate change, because as we've seen this year, even with the 5-year drought, I believe, in the Horn of Africa, thousands of livestock are dying, and that is affecting millions of people. And so we really need to improve our investment and attention towards these possible solutions with phenotyping technologies and genome editing, so that we can improve food security and help millions of people that are facing climate change right now.
Just a quick point as well. I think we were discussing this earlier as well. The communication of science is a huge aspect in this, especially in terms of genome editing, but also for phenotyping. I think the education part of it, especially in low to middle income countries, like reaching out to farmers, being able to communicate the importance of the science behind implementing these technologies for both genome editing and phenotyping is essential.
Lastly, I can just comment that I think the science behind gene editing is so cool, and it's very important. And it's just a passion of mine. I have the sciencey side brain, and the policy side was something I just learned doing this project with my colleagues here. So it was kind of interesting to put in perspective that not everything has to be about the advances in science. And that's what I caught myself learning. And so basically with science, we can keep moving forward, but we need to be careful and not get too ahead of ourselves.
Okay, I think we'll close that, based on the time. And unfortunately, our director will not be able to, break away from his meeting to attend today. So I'll just try to give some closure to the meeting. There's a general consensus globally that transformation of our agri-food systems is really needed to ensure that we, everyone has food and nutrition security, that the planet, environment is maintained sustainably, and all this is done in an equitable manner. That, of course, implies livestock as well, because livestock is an important agri-food system. Clearly, this transformation will rely a lot on new innovations to occur. One benefit about one, optimistic aspect is that we need new ideas, new enthusiasm, and I'm proud to see youth will be taking a big role in this transformation. So I just want to congratulate you for your presentation today, your thoughts that you've shared. I'd also like to thank Stephen and Elizabeth Lonergan for their support to you, for Shelley Taylor, who's directing the overall Global Studies program, and also Carmen Bain for her role in bringing academic innovation to the university. So with that, I'd like to wish you, safe return to Rome. I mean, to Iowa State, to the U.S. This weekend, and also more than that, wish you all a very bright future. And I look forward to following your careers and the impact you have on the world in the future. So, thank you. And I also thank all the participants and the technician that helped with this, with the smooth operation of this webinar. So thank you very much.